Welcome to the network of core facilities of the Institute of Microbiology, Bioenergies and Biotechnology of Aix-Marseille University, dedicated to life sciences. It includes outstanding platforms with state-of-the-art techniques used in biomolecular interaction studies. Firstly, we can screen for interacting biomolecules. Secondly, we can confirm their interaction through in vitro assays. And thirdly, we can try to understand their modes of interaction using spectroscopic methods. Some of our platforms have acquired local or national accreditations and are part of European consortia.
Sometimes people come with a lot of targets. On top of that, they want to identify or confirm protein or DNA partners. In that case, we use our high-throughput protein-protein or protein-DNA interaction assays on our robots to confirm or identify their partners.
We have several screening techniques depending on the scientific question asked. We have developed high-throughput screening methods to detect and quantify protein-protein interaction called the hold-up, or protein-DNA interaction using CellX. In addition, we use high-content screening of bacteria to identify new antibacterial agents. In a complex mixture, we can screen the proteome for interactants using LC-MS. Within supramolecular complexes, we can identify the partners and their stoichiometry through Edman sequencing. We have several techniques to detect the presence of interaction between two molecules. We can observe ligand-analyte interactions depending on the analyte type and size. We can detect interactions in volumes ranging from 4 microliters to 250 microliters, with binding affinities down to the nanomolar range on supports or in solution.
For my PhD project, I use interferometry. I use Octet N1 when I need fast check of my multiple batch of vesicles. It permits me to use a small amount of vesicles. I use also Octet Red, which is more powerful and have 8 heads. It allows me to do simultaneous jobs and more precise calculation to achieve KD and to compete different antibodies against my vesicles.
The ITC technique can detect interactions of proteins with a myriad of analytes, metals, carbohydrates, protein and DNA. Using intrinsic fluorophores or fluorescent labels, we can detect interactions down to picomolar range on the monolith X. We can also detect the oligomerization effect and conformational changes in proteins due to interactants by sequels. How do the molecules interact, and where does the interaction take place within the molecules? Using the ITC, we can determine the thermodynamic parameters of the interaction and infer which type of binding mechanism is involved and the stoichiometry of the binding. We can also identify the regions involved in the interaction by NMR and EPR.
Using EPR, with 1 or 2 paramagnetic inserted probes, we can detect any structural transition occurring during the interaction. And we can also identify the residues involved in this interaction.
In addition, EPR can detect the effect of substrate on the metallic centers in metalloenzymes through changes in the magnetic properties. Thank you for watching this video, which highlights our strengths in biomolecular interactions. If you are interested in our services, in collaboration, innovative projects or training, please visit our lab website and contact us. More details about protein production and structural analysis can be found in the other IM2B dedicated videos.